The inside of a membrane, right here, we have all the, the, the phospholipid heads. Right up at the top, you can see this extracellular, outside the cell, extracellular space. This is our phospholipid. See, one layer, two layers. So one, two. So we call it bilayer. You can see it written there. And this right here is a cytoplasm. Inside the cell right there. And it's important to understand the phospholipid head is hydrophilic, and the tail is hydrophobic. And this is really the key of what makes the membrane work. You have a waterproof layer, allows the cell to control what enters and leaves. Okay, it can just seal out most of the environments. The life involves water, so by having a waterproof layer, you can control entry and exit to your cell. So we got a lot of membrane stuck inside the membrane on the outside and through the middle, and lots of different jobs. Hormone binding sites. I'm sure you've heard of insulin. Insulin can join and then create a signal, tell the cell to move sugar in. Enzymes can be stuck on the membrane, so they can work, and your small intestine has a lot of these to digest your food. Cell adhesion. Cells need to stick together, just like IB students. And in all your tissues and your organs, where they rely on that cell-to-cell communication. Cells can basically talk to each other. I mentioned before, while you want to create a barrier, you have to get stuff through it. So you have channels for substance to move through. Aquaporin is one, right over here. Aquaporin, and you might guess aquaporin, what it's used for. It's used for water. And pumps for active transport, and we'll get to this later. Sodium, potassium pumps, active transport using ATP. And this next slide, you got a nice little diagram showing all this. So between cell, you join together. Intercellular, so between the cells. These are the enzymes that are stuck in there. So enzymatic activity. Transport. You have both. This is passive right here, and this is active. See this ATP being used, pushing things against their concentration gradient. Passive, things go along their concentration gradient. So basically high H to low. This is going here, you're going low to high, which is like going uphill. Here, cell to cell recognition. You see we have anchorage and attachment. And then signals. We talked about earlier, this could be insulin making contact with your cell, and then your cell then accepting, doing some action. Okay. Here, another really, I think, a pretty cool example or graphic of integral proteins. You can see here's the two layers, and these proteins going all the way through one side to the other. A recognition receptor, and this thing called a helical bundle for enzymes. A P barrel. Check this out. Look how cool that is. All these beta pleated sheets going through, and then wrapping all the way through the membrane. And then things like enzymes anchoring peripheral proteins. You see, they're just on one side or the other, either outside or inside. So this is the basic cholesterol. It's a lipid, and it has this basic shape, A, B, C, D. So I just want just to notice the same basic shape, but you have different structure. So this is different. The structure this progesterone has is different from cholesterol. Testosterone has this one OH group. Cortisol, look at this OH group coming off of here that nobody else seems to have. Oh, and aldosterone right here. That's unique. So those are some different things. I just want to make a note of that. Okay, so here's your basic model. Here's a glycolipid. Go back here. Glyco means sugar, lipid meaning fat, and here showing the phospholipid. You know, the head and then the tails right there. Cholesterol, this little chunk here. Normally we think of cholesterol in negative terms, but we need cholesterol. Oh, alpha helix protein. I think I have yet another. So that skipped that. I'll have to go back and edit that. But here, draw your own. You can use the previous diagram or the one in your book as a model, but get your phospholipid bilayer all set up with the phospholipid heads and the tails. You got your peripheral proteins. I got those labeled. You got your integral or transmembrane protein phospholipids. Cholesterol, have right there. Carbohydrate, glycoprotein. So just draw that yourself. And so here's the two major models. The first one, 35, and then the one we currently use, the Singer-Nicholson. We had in the first two previous slides. You notice the main difference here, the protein on the outside. I did a bad job with that. So protein, a layer on the inside and on the outside, and then no proteins through the middle. So that's the difference. You know, protein layers on outside with this Davson-Danieli model. Here, our current model, we got protein interspersed throughout. And the evidence why they first believed this, because they had this, and proteins look dark, electron micrographs. So they saw this, the light is phospholipid. So they saw it looked like two railroad tracks. Oh, it looks like a dense layer of proteins on the outside and on the inside, and then the phospholipids in the middle. So came up with their model. But then they started to have some things show up as to refute that one. And one thing was this freeze etch electron micrographs. They saw a real globular shape. Globular, dense, you know, in the freeze etch, showing that some proteins. Having trouble with my thing here. Some went into the phospholipid layer. Okay, so a lot of dense, showing some. It wasn't just smooth like they would expect. So that doesn't quite fit. Oh, let's get out of here, bug. That doesn't quite fit the model. You have these bumps and everything, and there's a picture either here or in your book. The other thing, they were able to isolate the structures. So they isolated the proteins. It's amazing what they can do in molecular biology. They isolate the proteins, and they found they were very different Very different shapes were involved. And if you're forming a layer on the outside, you're going to want something pretty uniform, but that's not what they found. All these different shapes of protein, sizes, all sorts of different stuff. So that didn't really fit the model either. Also, some were hydrophobic. And you know what that means? Hydrophobic, going in the middle of the membrane, it's hydrophobic. So they shouldn't be able to go in the middle if they're hydrophobic. But since they are, they must be integrated into that protein. And probably the coolest thing, they did this fluorescent antibody tagging. So they took a cell, they put all these, say, green fluorescent tag proteins here. They took all these purple fluorescent tags here, and then they just see what would happen over time. So I don't know exactly how long it was, 20 minutes, an hour, a day, I don't know. I wish I did. Eventually, guess what they found? Not too much time, they found these proteins were mixed. So they mixed. So it doesn't really fit that idea, as they're just kind of this layer encasing the protein. They're integrated, they're moving, they're very active. And there's another pretty cool image here. This is showing the freeze etch technique, kind of splitting the membrane right down the middle, and you see these proteins pop out, all these bumps you can see on the actual etch. Pretty cool, you know, to have a model but actually see, kind of like we did today in class when we looked at, I had you guys identify different organelles and electron micrographs. So this is basically what I just drew. This shows that membrane proteins move, do not form solid layer, you know, inside and outside. So showing that activity. Eventually they were fused together, these two different cells, and then they eventually all mixed together.